So from the science perspective today, we have Dr. JPaul from the University of South Florida. And John specializes in marine microbiology. His recent focus has been on the molecular detection of harmful algal blooms, bacteria and viruses, as well as the detection of seafood fraud and mislabeling. And in 2014, John started a company called Pure Molecular LLC with his former graduate student, Dr. Robert Earlrich, to develop assays for handheld genetic identification technology. This is a major step forward for combating seafood fraud. So we look forward to hearing from you, John, on that topic. From the industry perspective, we have Bill Dewey, who's the Director of Public Affairs for Taylor Shellfish, which is one of the largest producers of farmed shellfish in the US. Since receiving his degree in shellfish biology from the University of Washington, Bill has worked as a shellfish farmer in Washington State. So in addition to his role at Taylor Shellfish Farms, Bill also owns and operates his own Manila clam farm and serves on a number of boards and committees related to shellfish aquaculture. We also have Peter Koufopoulos here, who's going to bring the agency viewpoint from the Food and Drug Administration, from FDA. Since 2015, Peter has been the director of the Division of Seafood Safety at FDA's Center for Food Safety and Applied Nutrition. Peter has been with FDA since 2001, serving as the chief over seafood HACCP, which is the Hazard Analysis Critical Control Point, and working in the New England District Office as both a regional shellfish specialist and an investigations supervisor. So I'm very pleased to have all of you here today to speak on your various expertise of topics on food safety. So I'm going to go ahead and turn this over to JPaul. Thank you, John.
Thank you. I want to thank the Ocean Consortium for inviting us to talk today about some of the work that we have going, and I'm sort of going to set a general background on what we know about food safety and seafood safety. Food safety and security. I came upon this image as sort of a Venn diagram that shows the relationship between food safety and food security. Food safety is governed by issues in raw materials, pathogens, processing, cross-contamination, and one of the things I tried to white out at the bottom was climate change, but it kind of went through the white blockage there. Then there's our food security issues on the right side, and there's an overlap with poverty and climate change if you believe in climate change. But the point is, critical issues on Earth's populations reaching 10 billion. What role will seafood play in maintaining food security in our nation? And food safety is an umbrella term that encompasses many facets of handling, preparation, storage of food to prevent illness and injury. Included are chemical, microphysical, microbiological aspects of food safety. Pathogenic bacteria, viruses, and toxins produced by microorganisms are all possible contaminants of food and impact food safety. Allergens can cause 200 deaths per year and numerous trips to the emergency room. Foreign objects in the food, which we don't hear much about, constitute microphysical dangers, including bone chips, metal flakes, fragments, and shotgun pellets, actually. Microbiological food safety, about 9.4 million people are sickened and 1,350 deaths occur each year in the United States due to 31 major pathogens that contaminate food. Therefore, microbiological quality is the most important aspect of food safety, arguably. Norovirus, Salmonella, Clostridium, and Campylobacteria are the leading causes of foodborne illness, and foodborne deaths are primarily caused by Listeria and E. coli 0157H7. And the illness occurrences, I was surprised to see norovirus is number one with 5 million cases. 58% of the illnesses occurred in norovirus. Norovirus is the cruise ship virus projectile vomiting thing that goes through you in 24 hours and really incapacitates you. And then we see Staph aureus is the fifth highest, and something we have interest in from another perspective. Now, seafood safety, 95% of the seafood consumed in the U.S. is imported from foreign countries, and 1 to 2% is inspected by U.S. agencies. The rationale for inspection is defined by the agency's mission. There's a lot of overlapping activity in inspection. The FDA is concerned with the health and safety of seafood consumption, that people don't get sick from consuming seafood. NOAA is concerned with the fishery sustainability and stock assessment and how this can be maintained in a good fishing pressure. And U.S. Customs is concerned with payment of duties and tariffs, and that's the motivation for their inspection. Now, seafood fraud, the Oceana NGO reports of 2013 2015 really got people awake to the occurrence of mislabeling, and on the average, 30% of the finfish consumed is unintentionally or intentionally mislabeled. The most mislabeled fish was red snapper at 87%. 87% of the time you order red snapper, you're getting something else. So that's kind of scary. Tuna second at 59%. So this has led to a seafood black market that's valued at $25 billion annually. Now, what are the health effects of seafood mislabeling? Well, allergies. People are allergic to certain fish and things that are mislabeled, and they don't think they're getting something that's going to kick in their allergy, and in fact it does. Toxin and non-metabolizable compounds. Escalar is served as white tuna. Heavy metals, pesticides, human waste in the production of fisheries. And shrimp, the largest seafood industry in tons landed, is misrepresented by 30% of the time. What do we mean by misrepresenting shrimp is that farm-raised shrimp is misidentified as wild caught, and there's quite a difference in the safety of wild caught versus farm-raised in Southeast Asia, and we have to be aware of the actual source of our shrimp. Now, science and technology in seafood safety, I'm going to talk about three little areas. The detection of microbial pathogens in seafood, what I call seafood forensics, detecting mislabeled seafood, and then the detection of harmful algal bloom toxins in seafood and shellfish. Now, in terms of detecting microbial pathogens in seafood, there are rapid methods to specifically detect pathogens like Vibrio vulnificus, Clostridium botulinum, Listeria, and others. There's got to be a need to discriminate between the virulent versus harmless microbes that are to identify the environmental reserves of pathogens. For example, Vibrio cholerae doesn't always cause cholera unless it has the prophage genome in its genome. So we need to identify the actual pathogen disease-causing and sickening microbial species in our seafood. Now, in terms of seafood forensics, it's important to detect mislabeled seafood and where this occurs. Point of sale detection technology. Is this happening at the wholesalers? Is this happening when the imports come in from foreign countries, or does it happen actually in the restaurant or the seafood vendor? And the point of this is to restore consumer confidence in our seafood industry. detection of HABs and HAB toxins in seafood and shellfish. Here's quite a fish kill, and we see that right now of an area of extreme interest is the HABs in freshwater, in the Great Lakes. We have a real problem with microcystis and sliming of the water, as well as having neurotoxins. And this anatoxin is an extremely virulent toxin that's produced by Anabina and the Oscillatoria type species. Domoic acid is a neurotoxin produced by Pseudonychia, that's a diatom. Alexandrium produces saxitoxin, which is paralytic shellfish poisoning. And we in Florida have Carenia brevis, which is our Florida red tide organism, and that contains a neurotoxin. But as far as I know, nobody's been killed by Florida red tide yet, though it's made quite a few people sick. Now, what's the role of genetic technology in seafood safety? Well, right now there's two major technologies for identifying seafood, and this is nucleic acid amplification, or NAA, and this is a figure of the left-hand side of an amplification plot. It's a means of specifically detecting whether a species is present or absent. It doesn't tell you if the what the species is, as sequencing does. In sequencing, we sequence the barcode of life or several other genes, but the differences in the speed of these processes is that in 15 minutes to 2 hours you can detect if you've got the target organism you're looking for. Sequencing takes generally a day or two, so it's not quite as rapid in giving the information that we need if we're buying seafood and we want to know exactly what the species is. I bring in a shameless plug for my own company, Pure Molecular LLC. We developed a grouper check assay and what we call our tricorder. And it's so easy that you take a little plug of tissue and put it in an extraction reagent, put it in our little magic machine, and this can tell you if grouper has been detected, or if it's an imposter, you get a flat line. So this is the handheld technology that we've been employing in a lot of different arenas. The genetic methods enable rapid detection of microbial pathogen contamination in seafood. Novel methods enable rapid identification of seafood and limit mislabeling. One of the things we like to say is that nucleic acids don't lie. You get your answer, and it's a firm yes or a firm no. One of the things that we are dealing with is the increases in intensity and duration of harmful algal blooms worldwide, and we don't know if this is a temperature or pollution phenomena, but certain fisheries have been totally wiped out by harmful algal blooms. Chile has had a dreadful time in the past year. Virtually all their salmon hatcheries have been wiped out by an Alexandrium Caton alum bloom. What I didn't get to talk about was immunological methods to provide rapid and accurate harmful algal bloom detection technology. And this is where I was going to finish my talk. We have one planet, one code. We use the nucleic acids for seafood authentication, red tide detection, monitoring, bacterial pathogens, and viruses. So if you have one technology, you have the key to understanding all these different organisms that can interfere with seafood health and safety. Now, someone in my area in St. Pete said, well, you're going to Washington. I'd like to put in 2 quick slides. And this is the Magnuson-Stevens Fishery Conservation and Management Act Seafood Import Monitoring Program. This is the final rule by the National Oceanographic and Atmospheric Administration, published on December 9, 2016, designed to combat illegal, unreported, and unregulated IUU fishing or seafood fraud associated with imported seafood. Establishes permitting, reporting, and record-keeping procedures, including collection of catch, landing, and supply chain traceability for species. So the buzzword last year was traceability, and the reality of the situation is that it requires each vessel to trace back the input of much of the data for each, the vessel name, the flag, the license, the number, other identifiers, fishing gear, species, product description, quantity, weight, and bought, area of capture, harvest dates, point of land, and et cetera, et cetera. A single container of imported mahi that this company that we're talking about can have many different product forms, portions of various sizes, skin-on fillets, et cetera, and can potentially be made up to fish from 10 to 30 boats or more. So a single typical container of mahi, 38 to 44,000 pounds, can therefore require hundreds of discrete data point entries. So the traceability, although it's a good idea, in actuality it's quite a burdensome to the industry, and then once you have all this information, who looks at it? You know, you give all this information, and does it just end up in a file folder somewhere? So that with that, I'd like to end, and I guess questions at the end. Okay.